Hello everyone, this is Dr. Meenakshi Gupta. Today I'm going to start another topic of pharmacology, that is RAS. And if we expand it, it is a Renin Angiotensin Aldosterone system. So what is this system? It is a hormone system in the body for the regulation of blood pressure, then fluid, body fluid, and electrolyte balance. Now, what are all the involved for the regulation? The three things which are involved, they are, if we expand, they are renin, then angiotensin, and the third one is aldosterone. So all these, they gets activated in a sequence. Now how this system it gets activated? Okay? It gets activated whenever there is a suppose fall in blood pressure or there is decrease in a body fluid. Now what happens once there is a fall in blood pressure? There is a release of renin from the kidney. There is a release of the renin from the kidney from here. Now you all know in the kidney, the functional unit of the kidney, that is a nephron. So this is a nephron. Now you can see here in the nephron, this is the afferent arteriole, and this is the efferent arteriole. So this afferent arteriole, they form a network of the capillaries over here. This is called as a glomerulus. Now the blood comes from this afferent arterioles, moves towards this glomerulus. Then what will happen? This whole thing, it contracts. Okay. So what will happen? The filtrate that is produced this way. And around 120 to 125 ml per minute, that filtrate is produced, or nearly about, you can say, in a day 180 liter of the filtrate that is produced. So out of this 180 liter, mostly 99% that is reabsorbed throughout this tubule. The 99% of the filtrate that is reabsorbed, and the maximum reabsorption that happens in the proximal convoluted tubule, 65%. Okay, 15% over here, then 5 to 10% over here, and then 5 to 10% over here. So this way the reabsorption of the filtrate that will happen. Now what happens if there is a fall in blood pressure? Then the pressure in the glomerulus, that will be less, or renal perfusion, you can say, the renal perfusion will be decreased. Now once this renal perfusion through this arterioles, afferent arteriole, if this is decreased, then what will happen? The wall of this, it gets stretched because of the less perfusion. Here is a important granular cell in the afferent arteriole, which is a JG cell. Now they are going to sense this decreased renal perfusion, and what will happen from this granular cells or from the JG cell, the renin that is released. Okay, so renin is released, number one, because of the decreased renal perfusion. The JG cell gets stimulated, which causes the release of renin. This is number one. Number two, if the blood volume that is decreased, okay, or if there is a fall in sodium concentration, suppose if the blood volume that is decreased, then what will happen? This perfusion again it is decreased. Then to cover up that blood volume, from here this sodium will be reabsorbed. More amount of the sodium will be reabsorbed to cover that blood volume, because the sodium that is reabsorbed, along with that water also follows the same. Then what will happen? The concentration of this sodium in the DCT, here it is a distal convoluted tubule. So distal convoluted tubule, here the concentration of the sodium that is less. Now this concentration of the sodium is less. This is sensed by another group of cells, that is this green color I am showing. This is present in the distal convoluted tubule, and this is called macula densa. Now they are going to send the signal. They are connected with the JG cell with the help of connective tissue, and they are going to send the signal to this JG cell, and again the renin will be released. So second stimulus of renin release is the low sodium. This is going to stimulate the macula densa cell of DCT, and that is going to stimulate the JG cell, right? And renin will be released. Now number 3, the innervation of this JG cell. Which receptor is present? It is innervated by the sympathetic nervous system. Beta 1 receptor, they are there. So direct sympathetic stimulation. Number 3 is the direct sympathetic stimulation of the JG cell. That also leads to the renin release. So renin is released. What are all the 3 stimulus? Once, if the renal perfusion that is decreased, then it is going to stimulate the JG cell and renin will be released. Number 2, if there is low sodium because of the low perfusion, again, first one is because of the decreased blood pressure. Second one is again because of the decreased fluid. So low sodium will be sensed by the macula densa cell of the DCT, distal convoluted tubule, which is going to stimulate the JG cell, and renin will be released. Number 3 is direct sympathetic stimulation of the JG cell. So these are the 3 important stimuli which causes the release of the renin in the circulation. Now, once the renin it comes, then what will happen? Once the renin comes in the circulation, okay. Now this renin, it comes into the circulation. This is the blood vessels. Okay. Now this is going to act on another protein, that is angiotensinogen. Now you can make out by the figure only. It is alpha 2 globulin which is synthesized and secreted by the liver. So this renin, it is going to act on this protein, and what it is going to form? It is going to form angiotensin 1. Now this angiotensinogen, its synthesis can be increased, or it is increased by a few hormones like glucocorticoid. They can increase estrogen, they can increase, and they say even the mutation, the gene which regulates its synthesis, the mutation in that gene, it is found in hypertensive patient. Now this angiotensin 1, it is inactive as such. It does not have any much action, and immediately it is cleaved by another enzyme, that is ACE, angiotensin converting enzyme. So another name for angiotensin converting enzyme is kinase 2. It is one and same thing, and this is going to convert this one angiotensin 1 to angiotensin 2, and this is active. It is going to exert many action by activating the receptor, that is angiotensin 1. See, there are two type of receptor, AT1 and AT2. So they are a little bit confusing. So many students, they think this AT1, that is for the angiotensin 1, and AT2, it is for the angiotensin 2. Both the receptor, they are for the angiotensin 2. If we expand this AT1, it is angio, A stands for angiotensin 2 type 1 receptor. Here also angiotensin 2 type 2 receptor. So both the receptor, they are for the angiotensin 2. And most of the action of angiotensin 2, they are exerted by angiotensin AT1 receptor. Okay? So maximum action that is exerted by AT1 receptor. And then right here again. And AT2 receptor, it is mainly present in the fetal tissue and in a few other like you can say mainly in the fetal tissue, and in the adult you can find it out in the kidney, in the adrenal medulla, in the kidney, in the adrenals, the liver. So these are the few sites of AT2 receptor, whereas AT1 receptor, now this angiotensin 1, it is going to act on AT1 receptor, and it is going to produce what all action by activating the AT1 receptor. So number one is it is going to cause a generalized vasoconstriction. I'll use another slide. Okay. So this AT1, it is going to act on the blood vessels and causes a generalized vasoconstriction, which is going to cause the rising blood pressure. Then another important site of this one is it is also going to act on this, the adrenal gland, on the adrenal medulla. This AT1 receptor, they are there, and adrenal cortex. By activating this adrenal medulla, it is going to cause the release of, on adrenal medulla, there will be the release of both epinephrine and norepinephrine. Then on the cortex, adrenal cortex, there will be secretion of aldosterone. This also causes a vasoconstriction. So there will be rising blood pressure. Then it also acts on the posterior pituitary and causes the release of ADH. So this and this aldosterone and ADH, their action is, both of them, they cause sodium and water retention. So this is going to increase the blood volume, and there is a rise in blood pressure. So these are the action of angiotensin 2 by activating the receptor AT1. Now the half-life of this renin, it is 15 minutes, whereas the half-life of this angiotensin 2, it is a 1 minute. So this angiotensin 1, it is degraded into angiotensin 2, it is degraded into 3 and 4 by various amino peptidases. So they are also inactive. So only the angiotensin 3, it exerts the same action just like angiotensin 2. It also causes the release of the aldosterone. And these 3 and 4, they are further degraded by angiotensinases into the inactive product. So this is the wait. Now in the beginning I told you it is a hormone system. If you remember, it is a hormone system in the body for the regulation of the blood pressure, body fluid and the electrolyte balance. Now just like the hormones, so here what are the hormones? The three things. This is an enzyme. This is, all of you know, it is a hormone. And angiotensin 2, that is also a hormone. So another enzyme which participate in this system, that is a ACE, angiotensin converting enzyme. Now angiotensin 2, it is a local hormone, or you can say it is a autocoids. So what are the autocoids? Which is released by the tissue and it is going to act at their site of release. Okay. Now just like a hormone, there is a feedback mechanism. So feedback mechanism, how this renin, it is, once it is released, then what are all the mechanism which is going to inhibit the release of the renin? So there is a short-term negative feedback and a long loop or a long-term negative feedback mechanism by which the renin release that is inhibited. So long-term is whenever there is rise in blood pressure, all these three, they are going to decrease the release of renin. This is a long-term. Long-term negative feedback. And short-term negative feedback, what is there? The AT1 receptor, they are also present in the JG cell. This angiotensin 2, they are going to stimulate this AT1 receptor and this is going to inhibit the renin release. This is a short-term negative feedback. So two negative feedback they are there. One is a short term, ठीक है, एक आपका short term है, another one is a long term. Short term, by activating the AT1 receptor on the JG cell, it is going to inhibit the further release of renin, whereas a long term, it is going to inhibit the renin release once there is a rise in blood pressure. So this is about the circulating RAS. Okay, this is all about the circulating RAS. And there is another important is that is a local or tissue RAS. Now local or tissue RAS, the few books they say it is further of two, extrinsic and intrinsic. So extrinsic is what they say in the blood vessel here, in the intima of the blood vessel, what they will do, they are going to catch up this renin and angiotensinogen, and here what will happen, angiotensin 2 that is formed locally and it is going to exert the local action. Then in the intrinsic, so few tissues, important tissues, they are heart, then blood vessels, kidney adrenal, brain, liver. They have also this local RAS. Now here, the renin in the circulating, it comes from the JG cell of the kidney. Whereas in local RAS, renin, it is synthesized as a larger peptide, that is pre-pro-renin. Okay. As a pre-pro-renin, which gets activated with the appropriate stimuli into prorenin and renin, and both are active. It is not like that the prorenin, this is inactive and this is, both of them, they are active form, and rather this is amount of this one is a more 5 to 10 fold as compared to the renin. Now this prorenin and renin, now both of them, they are active. So they are going to activate the angiotensinogen by two ways. One is enzymatic and non-enzymatic activation. So non-enzymatic activation, that is with the help of prorenin receptor. This prorenin receptor, this helps in the non-enzymatic activation. Now enzymatic activation, how this is going to happen for the prorenin, you can see in the next figure. This I have taken from the Tripathi. So this is a prorenin. So enzymatic activation, that is with the help of proteases. Here the proteases are proconvertase and cathepsin. So what they do, they are going to remove this propeptide, which is hindering the activation of this prorenin. So what will happen? They are going to cleave off this. You can see here. Then what will happen? This prorenin, it gets activated into the renin. This active site of this one, this active site of this enzyme, it is exposed. So angiotensinogen, it comes and bind here, and resulting product, the angiotensinogen, that is converted into angiotensin 1. So this is a enzymatic activation of the prorenin. Now non-enzymatic activation of the prorenin, here what happens? In a non-enzymatic activation, the prorenin receptor, they act as a scaffolding. This prorenin, it binds with the receptor. Once it binds with the receptor, then what will happen? The conformational change on this prorenin, it happens. With the conformational change, this inactive propeptide, you can see the conformational change. Once the conformational change happens, then this prorenin, which was hindering here for the action, so you can see the change, and this catalytic site, that is exposed, and angiotensinogen, it binds with the catalytic site and it gets converted into angiotensin 1. And if it is a prorenin, it causes the activation, and if it is a renin, it causes the augmentation of its action. So non-enzymatic and enzymatic. So this one is a local or tissue RAS. So where, how the renin, it gets activated, enzymatic or non-enzymatic. So non-enzymatic, that is the main pathway for the local RAS, and they are going to produce many action by two pathway. It may be angiotensin 2 dependent pathway or angiotensin 2 independent pathway. So this illustration I have taken from the KD Tripathi. Even the next illustration, this is also I have taken from the KD Tripathi. So here you can see this non-enzymatic pathway, the same, the previous, you can see this non-enzymatic pathway where this prorenin receptor, here prorenin receptor with the prorenin receptor with this prorenin binds, and renin augmentation of this and activation of this happens. So non-enzymatic activation, it is maybe angiotensin 2 dependent, it may be angiotensin 2 independent. So angiotensin 2 dependent pathway, here what happens, angiotensin 1 that is formed, and this angiotensin 1 it is converted into angiotensin 2, and then further action that is produced. Whereas angiotensin 2 independent pathway, what happens here? So this is going to trigger angiotensin independent pathway. Here angiotensin 2 that is not produced, so they are going to trigger the intracellular signaling via the activation of this MAP kinase, PAI1, JAK-STAT, and all these they are going to cause the activation of various proto-oncogenes, transcription factor, and this all leads to cell growth. You can see fibrosis, apoptosis, hypertrophy, ventricular hypertrophy, myocardial remodeling, remodeling of the vascular muscle mass, nephropathy and retinopathy. So you can see the local RAS. So we are talking about the local RAS, local renin angiotensin system. It is going to cause all that. So activation of this local RAS leads to many pathological conditions, especially this vascular remodeling. Okay. Then we have a ventricular hypertrophy, myocardial remodeling, fibrosis, nephropathy and retinopathy. So in today's video I have discussed the renin angiotensin aldosterone system. I have discussed the two pathway, one is a circulating and another one is a local. The circulating pathway RAS that gets activated whenever there is a rise in blood pressure. So that is a one system which is going to regulate the blood pressure of the body. It is going to take care of both short-term regulation of the blood pressure and a long-term. Otherwise, the short-term regulation of the blood pressure that is also taken care of by the baroreceptor reflex. And another one is I have discussed the local RAS. So for all that, whatever is required for the RAS, it is also present in a few tissue like heart, blood vessel, kidney, adrenal, brain. Okay, in all that it is there, but how the renin it is produced in all these local tissue, that is synthesized as a larger peptide pre-pro-renin, and this pre-pro-renin with the appropriate stimuli it gets activated. There is enzymatic activation, you can see here there is enzymatic activation and there is a non-enzymatic activation. So non-enzymatic activation, that is with the help of pro-renin receptor, and that is the main pathway for the local RAS. And once it gets activated, then what will happen? So अब ये आपका angiotensin 1 it is formed in both way. Then further activation it may be angiotensin 1 2 dependent or angiotensin 2 independent. So in the next slide I have shown. So ultimately what it causes, it is going to cause all these activation of this local RAS. It leads to various pathological conditions which have been mentioned here. So that's all about today's video. If you like my video, please give a big thumbs up and share my video and subscribe my channel.